Okay, ready. So I want to tell you about stem cells and tissue regeneration. Piero asked me to cover the whole field, plus talk about the highlights of my own work, which I'll try to do in 20 minutes. A tall order. So let's see. So regenerative medicine is largely viewed as a solution to aging. And as you see in Monk's picture here, aging is often depicted as a young, vibrant child, and then a wonderful, full-bloom, middle-aged woman, and then she gets older, and she ends up decrepit and debilitated. And the question is, do we really have to age that way? And our reasoning with regenerative medicine is no. And as depicted in nature, it's been shown that our life expectancy is increasing both for men and for women, but as the article says, more years of what? And that is because our health span, the healthy years, are not increasing. These are more years of chronic disease and debility. So the goal of regenerative medicine is to counter that. So the idea is to increase health span so that as you age, you can do all the things you love to do, dance and run and ski, like this woman shown here. But regenerative medicine isn't just for aging, it's also for the young and for various life-threatening disorders. So the goals of stem cells and tissue regeneration are to regenerate or replace damaged or diseased tissues, for instance, for treating Parkinson's or heart attack or stroke, and to use cells to deliver genes that are missing in inherited diseases like muscular dystrophy or hemophilia, and to understand early human development and model disease processes, test drugs, as you probably heard about disease in a dish, and I'll describe that in more detail. But first I want to describe the three types of stem cells so that you all are, when you read about these things, you know what they are. Embryonic, induced pluripotent, and adult stem cells. So embryonic stem cells come from the early embryo at the stage where there's an inner cell mass. And these cells are then isolated, so shortly after a few cell divisions, and they're placed in culture. These are embryonic stem cells, and they can be differentiated into all different kinds of cell types, a large variety. The advantages of embryonic stem cells are that they undergo extensive proliferation, essentially unlimited as a cell source, and they can be differentiated into virtually any cell type. The disadvantages are, of course, that you're dissociating the inner cell mass, which involves the destruction of an embryo created by IVF. And there are ethical concerns regarding egg donors for in vitro fertilization for this purpose. And it's often more difficult than expected to direct these cells to become mature cell types. They often only express fetal genes. Moreover, they can become tumorigenic. So I entered this field in the 80s, as Kurt mentioned. I wanted to understand how a cell could be reprogrammed and whether a cell could be reprogrammed. At the time, it was thought that once a liver cell, always a liver cell, it couldn't be changed. And I wanted to challenge that idea. So what I did was to fuse mouse muscle cells with human hepatocytes into multinucleate syncytia. And the reason for that is that I wanted to bias the number of nuclei, or the gene dosage, to favor the muscle and see if I couldn't turn on muscle genes in a human liver cell. And the reason for having a non-dividing system was that in the course of cell division, it was known that chromosomes are lost and rearranged, and it's hard to know whether you activated a gene or whether you simply lost a repressor of that gene. I wanted to prove that there were transactivators that could induce new gene expression and test whether the destiny of a skin cell or a liver cell was terminal or could be reversed. And as shown here, we found that you could. So here's a mouse muscle nucleus and a human liver nucleus, and you see expression here of human muscle NCAM that this cell normally never would make. show this for a number of different genes. And this had to be due to transactivators in the, elaborated by the muscle cell that could activate these genes in the liver cell. So this showed that without chromosome loss or rearrangement, reprogramming and activation of silent genes was possible, and that the differentiated state is not fixed and irreversible, and that there's remarkable plasticity in the differentiated state. So others have also shown this in another way. Nicolas Durand was transplanting parts of the neural crest to different sites and found that they could be instructed by their microenvironment. But all of this led, more recently, to the discovery by Shinya Yamanaka of induced pluripotent cells. And that's what you hear a lot about. And in fact, it was predicated on some of the work, like my own, that showed that an abundance of transcription factors could alter cell fate and induce silent genes. And what he did was an incredible experiment, actually, where he took libraries of factors and screened them. And he, instead of taking a pool and you know, if you got a hit, then fractionating that into, say, 5 pools, and then the one that gave a hit, fractionating that further, he would never have gotten the result that he did. He did a brute force experiment where you had all the factors and then removed one at a time, and did that for every pool so that he always had everything represented. And when he did that, he discovered 4 transcription factors, which he expressed in episomal vectors, that if you overexpress them in the cells of your skin or urine or blood, you can turn on genes and make a cell that is like an embryonic stem cell. So it's quite remarkable. And then you can differentiate these, as shown here on this appetizer platter. I love this image. Myoblasts and neurons and endothelial cells of the vasculature and cardiomyocytes, whatever you like. You can also genetically engineer them and correct them. So the advantages of this technology is that you have an unlimited cell source like embryonic stem cells, and you can make them autologous. You could take a person's own, so you don't want to take a person's piece of their own heart, but you could take the blood and then reprogram it to be heart. And so you can have autologous cells with no immune mismatch, and it bypasses the use of embryos. And you can study disease mechanisms and perform drug screens, also tissue replacement. The disadvantages are that it's difficult sometimes, as with the embryonic stem cells, to get them to mature beyond a fetal stage, and there is a high risk of tumorigenicity. They are unstable. So, but this shows you what you can do, and we've done this in my own lab. We have induced these cells to become cardiomyocytes, beating cardiomyocytes, and we can get them to perform really well if we produce a hydrogel, a biomaterial on which we can align them. So what we do is we pattern the biomaterial so that we have an aspect ratio of 1:7, which is exactly what the cardiomyocytes like, and then they will align and you get these nice regular contractile apparatus. And now you can also embed beads, these fluorescent beads, in the hydrogel, and you can measure the placement of those beads, as shown here by traction force microscopy. And you can get a measure of the strength of output of the cells. And if you have cells that you derive from someone with a human genetic disease like Duchenne muscular dystrophy, where there's a missing gene for the protein dystrophin, which is key to the contractile, you can measure the arrhythmias, and you can then test drugs and see to what extent you can correct this kind of arrhythmia. So this is the notion of disease in a dish, where you can really get a nice phenotype. Now you can screen for drugs to correct it, you can characterize it, you can find out the mechanisms that have gone awry in a way that would be impossible otherwise. For instance, if you isolate the cells, the cardiomyocytes from a mouse, they survive in culture for maybe 1 to 2 days, and they're on their process of dying. And these you can monitor for quite a while. So we characterize these from Duchenne muscular dystrophy, which is due to a deficiency of dystrophin, from troponin T deficiency, and from titin deficiency. These are all structural proteins that are crucial to the function of skeletal muscle and also to the heart. And what we found was quite surprising, which is that as the, not only did we see arrhythmias, but we found that telomeres are shortened in the cardiomyocytes, specifically in these genetic dilated cardiomyopathies that lead to death. So they are premature diseases of aging of the heart, which is really was not previously known. And we characterized that first by looking at cardiomyocytes in the tissues of the heart from these patients. And then we were able to recapitulate this. Shows you again, disease in a dish, because we were able to recapitulate this with iPS-derived cardiomyocytes. So we made induced pluripotent cells. They all had similar telomere lengths. And here you see the Duchenne. They start out same telomere lengths, and then the telomeres drop to 50% in each of these cases. So it's contraction in the absence of. And what is remarkable about this, you may have heard of telomere shortening as a function of age, and it's always associated with cell division. And for instance, the cells in your blood, as you age, your telomeres will get shorter. But those cells have undergone a lot of division. What is amazing here is that this shortening is happening independent of cell division. So it implies a completely different mechanism of telomere shortening. And it also suggests ways that we are looking at now, screening for drugs and ways to intervene to stop the telomere shortening as a way to intervene in dilated cardiomyopathy. So people are thinking of CRISPR to correct genes, but that will be tailor-made to each patient. This would be something that might be more universal. So the third type of stem cell are the adult tissue-specific stem cells. So I told you about the induced pluripotent cells and embryonic stem cells. But these cells are resident in your tissues. So for instance, in your muscles, you have stem cells that are there ready to spring into action if there's damage and they're needed to repair the damage. You have them in your skin as well. And these cells are dedicated to repair that tissue. And various ideas are to either isolate cells, grow them up, and then give them back as a cell therapy. And now a new approach is to try and stimulate the cells that are present in your tissues already and get them to perform better. So for instance, probably the best characterized cell is the hematopoietic stem cell, which has been isolated, and a single cell can reconstitute all the cells of the blood. And that, of course, is very useful in the treatment of leukemias and lymphomas. In the cornea of the eye, also tremendous progress has been made in isolating the limbal stem cells. Say if someone had a burn injury, chemical injury, or physical injury, then their own cells can be isolated and grown in culture and then replaced and restored sight. There's been tremendous success with that. And with retinal pigment epithelial cells also in the treatment of macular degeneration, which is degeneration here of the retinal epithelial cells, one can grow cells from the unaffected area and then replace them quite effectively. This is still in experimental stages. The cornea is actually in the clinic. And then I think one of the big excitements this year was a report from in Italy, where they have been able to grow these adult stem cells that are epidermal, and they found a way to isolate the key stem cell that can self-renew and maintain itself as a stem cell long-term in culture. And that enabled them then to treat a boy with epidermolysis bullosa, which is a severe blistering disease with chronic ulcers. It's extremely debilitating. The kids can't do anything. Every time they rub something, their skin comes off. He replaced 80% of the skin on a 7-year-old boy, and the boy is playing soccer 3 years later. So it's a wonderful success story in isolating the correct stem cell that has stem cell properties, finding a way to culture it and then deliver it and repair the damage. So this gives you an idea of the prospects of the field. Cardiomyocytes, unfortunately, we have not yet found the cardiac stem cell. There have been reports, but they've not held up. And so people have been injecting all kinds of things into the heart, and for a while they have some positive effects. They don't do any harm. They have some paracrine effects. They secrete something that is beneficial by being there. And recently people have made cardiomyocytes from embryonic stem cells and induced pluripotent cells, transplanted them. And in monkeys, they've gotten them to function, but they still have arrhythmia. So it turns out they're going to have to specialize those cells, make atrial or ventricular, and not have mixtures in order to overcome this. So the heart still remains a challenge. What I work on is the skeletal muscle, and there are stem cells that reside in the skeletal muscle just beneath the basement lamina. Here you see one, the hallmark transcription factor. 40% of your body mass is muscle, and muscles atrophy with aging, and that severely impacts quality of life as we age. And so one of the things we want to do is change that. When there's damage, these cells are activated and they proliferate and can get fused into pre-existing muscle fibers. So we've done work over the decades on this, but recently we came up with something that looks quite exciting. We reasoned that this wave of inflammation, that's the first thing that happens after there's an injury to muscle, might be key to this stem cell activation. And so we did an in silico screen of stem cells isolated from the muscle just after injury, and we looked for inflammatory regulators, and we found EP4 was upregulated, and that's the receptor for prostaglandin E2. And what we found then, we looked at injured muscles and found that, lo and behold, prostaglandin E2 follows exactly that wave of inflammation. And we added it to isolated stem cells and saw this huge boost in numbers with just a very short exposure to PGE. So, and this shows you what PGE does to cells in culture. When you have the control from aged individuals, you see that they proliferate very little compared to the ones we treat with PGE2 when you look at them by time-lapse microscopy and follow the progeny. And what's happening is not only are they stimulating an earlier time to first division, so you get many more divisions, as you can see, but PGE2 is also protective against cell death, which is increasing in the controls. Remarkably, we found that this signaling pathway is absolutely required by the stem cells to function. If we genetically ablate the receptor, we get cell cycle arrest in culture. If we try to engraft the cells, they simply won't engraft. They absolutely need the PGE2 signaling pathway. And moreover, if we do a conditional knockout, genetically ablate the receptor on the stem cells and then do an injury and look 4 months later at their strength, what we find is that young mice actually get weaker. So this is so important that they really can't repair the damage in the absence of that receptor. And that's shown here for many mice by twitch force and tetanic force. Moreover, we tested it another way. We wanted to follow the stem cells, so we made a new mouse where you could read out endogenous stem cells by bioluminescence imaging. And after an injury, we gave them NSAIDs, non-steroidal anti-inflammatory agents like ibuprofen. And again, we saw a reduction in stem cells and a reduction in strength. And the New York Times picked up on this finding and said, bring on the exercise, but hold the painkillers. And there's a lesson there: no pain, no gain. Because if you take these anti-inflammatory agents like ibuprofen immediately after exercising in the gym or after you do a run, you're negating all the good you did, and you will get weaker. So, but the good news is, it also works to strengthen. So here we tried a different kind of damage. This is a damage from eccentric downhill running. The other damages were chemical toxin induced from snake venoms. We wanted to look at something more natural. And this is like downhill treadmill running. For 10 minutes a day, the mice ran downhill, and we gave them prostaglandin E2 for the first week. Second week, they ran again, and then 4 weeks later, we measured their strength. And we found a twofold increase in twitch and tetanic forces, and this is for a large number of mice. So this short bout of PGE2 stimulation led to long-term strength increase, which we think is pretty exciting. So just to conclude, there are 3 types of stem cells with different characteristics and different relative strengths and utility. And why do we have so little funding? It would be entirely different if we could crude oil from these stem cells. So with that, I'll take questions.
Thank you, Helen.